Rihanna, thank you very much for the introduction. Thank you, Jackson, for the invitation. And good morning to all of you in the US and that side of the Atlantic, and good afternoon to those of you who are joining us from Europe or elsewhere. It's a pleasure to be here. So I'm going to be talking about another aspect of FC receptors, the IgG receptors, and we've been attempting to identify antibodies which are silenced with regard to that, and hopefully I can show you why. That's my slide. Okay. So you will know that antibodies are fantastic drugs for treating all sorts of different diseases, and naturally speaking, they engage with various receptors like complements, FC receptors, when they aggregate on the surface of cells. And there are numerous FC receptors which engage with IgG, and there's others which engage with other forms of immunoglobulin. But just very briefly, in humans, they fall into three classes, which we'll call FC gamma R1, R2, and R3. And within those classes, there are various subclasses with different function, different expression on different immune cells, and antibodies of different subclasses bind to these receptors in a diverse way. Some bind with higher affinity, some with lower affinity. But nearly all of them engage with the immune system to activate it in some way and to create things like phagocytosis, cell-mediated cytotoxicity, except for the FC gamma R2b, which has a cytoplasmic tail that's got this inhibitory motif attached to it, and that downregulates responses. So they're extremely important in the natural function of immunoglobulins. And when we use antibodies for treating cancer and other diseases, then we're exploiting those FC-mediated properties. But sometimes, of course, things go wrong. And there was, unfortunately, about 14, 15 years ago, a tragic example when healthy volunteers were treated with an antibody targeting CD28 on T cells, and it caused a devastating cytokine storm with very long-lasting effects for some of those unfortunate people. And that antibody had been designed with the aim of avoiding the binding to FC receptors. It was in fact a human IgG4 antibody, and it was thought by some people that that would not bind well to FC receptors and wouldn't cause these types of immune activation events, consequence of aggregating the antibody. But unfortunately, that was not the case. And so people have been searching for alternatives. We knew that this type of cytokine storm was a problem from the very first therapeutic monoclonal antibody, OKT3, a CD3 antibody, which of course has been used for many years for treating organ transplant rejection until it was eventually withdrawn largely because of the side effects. And if it wasn't for those side effects of cytokine release, anti-CD3 antibodies might well be used for treatment of many other diseases, immunological diseases, and we ourselves research the use of CD3 antibodies for treating type 1 diabetes, where it's a very powerful drug, but the side effects are still problematic. Well, there's a number of attempts over the years to eliminate the binding to FC receptors and eliminate these side effects by mutating the FC domain in different ways. Here's a structure of a wild-type human IgG1 as a starting point. And one of the earliest mutations was this leucine 234 changed to alanine and 235 changed to alanine. These mutations here resulted in reduced binding to the FC receptors, and many of these mutants have entered the clinic. Another way of changing the binding was by removing the carbohydrates, which is here in the normally in the center of the antibody. And antibodies which have been aglycosylated by changing the asparagine at 297 to something else also have reduced activity, but not eliminated with regard to binding FC receptors. Well, these mutations and many others have been tested, but they don't completely eliminate the problem. And in fact, that CD3 antibody we used for treating people with diabetes, that was an aglycosyl antibody. And others used antibodies like the LALA mutation. And in each case, they still were not sufficient to really prevent completely those cytokine release problems and thus the side effects. So we screened a lot of variants using some sort of structural intuition and just a lot of screening to try to find variants which completely eliminated. And I'll just show you some of these binding results. So here's the LALA antibody, and you'll see there's quite substantial binding here to the human Fc gamma R1, and likewise with the aglycosyl antibody. That also shows quite substantial binding in this Biocore assay. You see 500 units or so of binding. And here's one of the best variants that's been described in the literature, proprietary variants of Roche, and that's called LALA PG. It's a triple mutant, and you'll see there's a lower level of binding, about 50 units in this Biocore assay. So when we screened a lot of variants, we found some with even lower than that, and this is one of our best mAbsolve variants, which has got no detectable binding whatsoever to the Fc gamma R1. And in this next slide, I've lined it up against a whole range of published variants. There's the LALA PG, which was very low. Here's the LALA and the aglycosyl, and in between there are a number of other variants which have been made by different people and used in the clinic. And you'll see it's only really the mAbsolve variant which has completely eliminated the binding. And this result also applies to the other receptors. So you can see briefly here, there's the different receptors, Fc gamma R1, 2, and 3. And in each case, the mAbsolve variants showing no binding, whereas the LALA and the aglycosyl show low levels of binding to all of the receptors. Now, this low level of binding, this can translate to something quite a bit more substantial in cell-based assays. So we've looked at these different variants by using assays, a reporter-based assay for either ADCC using, for instance, Fc gamma R3, or for phagocytosis using the Fc gamma R1, Fc gamma R2. And here we have an engineered effector cell. It's a URCAT cell which has been engineered so that it gives a response when it encounters antibody which is aggregated on the surface of a target cell. And so this signal is easy to measure, and it's a very simple, straightforward assay, very good for screening purposes. And here's just a few of the advantages of these assays. The protocol is extremely simple, very quick, and involves very few steps. And numerous different receptors are available in this format, and so one can study a single receptor at a time. That's a problem with natural immune cells because they often express multiple receptors. Here we can look at just one at a time. And the assay is very reproducible. Here's some dose response curves. Dose response curves from 3 completely independent assays, which I superimposed on one another here. And you can see that they superimpose very well. It's very reproducible from day to day. So you get just the responses, increasing antibody concentration, increasing luminescence in the assay. And this is, we're using the Fc gamma R3 effector cells. You get the same results with the others. So using those Fc gamma R3 effector cells, I looked at a number of these variants, and you'll see the mAbsolve variance is very, very low, no detectable release, no detectable activity. The others are also pretty low. LALA is showing something clearly measurable, as are a few of the others, but they're mostly fairly low. But when we come to the Fc gamma R2, it's a slightly different picture, because although the mAbsolve is still obliterated, these other variants are showing up to 40% of the wild type activity. And when we come to Fc gamma R1, the next, yes, there's the Fc gamma R2b. That's very similar, and the Fc gamma R1. You see now that those modest levels, just 20% or so in binding activity, can translate to really substantial cell-based activity, except for LALA PG shows a little bit, and the mAbsolve is still completely eliminated. So this set of mutations that we have is something that really eliminates the binding very effectively. Does that translate to cytokine release? Yes, it does. So I've just got a few examples here from human peripheral blood mononuclear cells, and we've done a fairly conventional assay. In fact, this was done by our partners at Solentix, and they've looked at a whole panel of inflammatory cytokines. I've just showed a couple of them here. For example, interferon gamma, which gives a high level of release with a wild type antibody, and a low but still statistically significant level with the LALA and aglycosol variants, but the mAbsolve variant is indistinguishable from the buffer control. And you see the same thing over here with IL-2, mAbsolve indistinguishable from the buffer control, but now the LALA and aglycosol, as they do with some of the other cytokines, they come up to quite substantial levels. And so this mutation has eliminated the problem of cytokine release in this experiment with peripheral blood mononuclear cells. Now, an important factor is that we don't change the binding to FcRn or the half-life of the antibody by making mutations that knock out binding to the IgG receptors. And so here's a simple Biocore binding experiments where we've compared the binding of the mAbsolve variants with the wild type. So each time the mAbsolve is the dark blue, wild type is the light blue, and here we've got the LALA and the aglycosol variants tested as well. And in fact, most of these variants that I tested show rather little difference between the wild type and the variant antibodies. Of course, we weren't aiming to change the binding to FcRn. Here we've looked at FcRn from human, monkey, and mouse, and the results are similar with all of them. And in fact, these experiments have been repeated in another laboratory by my colleagues at Antibody Analytics, and they've done some rather more comprehensive Biocore experiments to actually measure the affinity by titrating the antibodies. And here's a few examples from their results. Here's the human FcRn with wild type immunoglobulin, and you'll see a KD of 562, human FcRn with a mAbsolve variant, and the KD is almost exactly the same. And they obtained the same results with mouse. Here's the KD 71 and 73 with the wild type and the mAbsolve variant, and in fact the same with monkey as well. I haven't put that up because it's just the same. Now, does that translate into pharmacokinetics in vivo? And this is, of course, where these mice, these transgenic mice that you've been hearing about from Jan's talk, come in. And we've been very happy, very pleased to be able to collaborate here with JAX Laboratory to do these experiments. And so we've done 4 different experiments in different types of mice. First of all, the wild type black 6 mouse, that's the background for the other strains. Of course, that's just carrying a mouse FcRn, and we'd hope that we would get similar results with the variant and the wild type because they do, unlike albumin, the human immunoglobulin does bind to the mouse FcRn. And then we've used the TG32 mouse. This one has got the mouse FcRn knocked out and the human FcRn inserted. That's the same mouse you just heard about. That's under the control of a native promoter, and so you get a rather natural level expression of the FcRn. A very new model that we've used is this TG32 with human IgG1 additionally inserted. And the reason for this is that the mouse immunoglobulin does not bind well to human FcRn. And so the TG32 mice, with their mouse FcRn knocked out, they have very low levels of endogenous immunoglobulin, endogenous mouse immunoglobulin, because it's not being rescued by the human FcRn. It doesn't bind to it. And that could potentially, we thought, be a complication for our studies, because that would mean we're injecting these rather small amounts, or tracer amounts of test sample into mice which are essentially deficient in immunoglobulin. And then that might slightly change the situation with regarding binding the mouse IgG receptors. So we thought it would be of interest to use this very new model, which is that much closer to humans because it's got human immunoglobulin, and therefore it's got closer to natural levels of circulating non-specific immunoglobulin in the mouse when you inject your test sample. And additionally, we used another mouse model, which is called TG276, and this has the human FcRn inserted under control of a ubiquitous promoter so that it's expressed as higher levels. And this strain is said to be, although it's not necessarily such a great or perfect model of the human situation, it's a good model for detecting small differences because it's said to be very sensitive to little differences in the binding. And so just briefly here, the results from these experiments. First of all, the wild-type black 6 mice. You see, we tested the wild-type antibody, that's in blue, the LALA variants, that's the well-known variants, in red, and the mAbsolve variant 252 in green. And you see the pharmacokinetics of all of those variants was very similar, with T-half in the region of 11 to 23 days. And in the TG32 mice, we got these. Now we've got the human FcRn, the same samples, and we'll see the same sort of result, that very close correlation with the T-halves in the range of 9 to 11 days for these three variants, the wild type, the LALA, and the mAbsolve variants. And then looking at the other mouse models, with the TG32 with carrying a human FC, it's expected that because the receptor is now saturated with natural normal human immunoglobulin made by the mouse, that the T-half will be shorter, and indeed it is. And again, we see rather similar results for all three variants. Now the T-half is about 4 to 5 days. But the results with the TG276 were a little bit different. And we don't see linear pharmacokinetics, which was a surprise to me. And I can't really understand quite why that is. This is under the unusual CAG promoter, and so it's expressed in different cell types, perhaps from normal at different levels. I'm not sure of the implication of that. And you'll see here that the wild type antibody has a longer half-life than the variants. In this case, we tested, as well as the LALA, we tested an aglycosol variant, that's in green here. And you'll see that all of the variants which had that reduced binding to IgG receptors show a slightly shorter half-life in that model, which is said to be very sensitive, but not necessarily replicative of the human situation. So just in summary, that's quite a short presentation, but we've got a few conclusions from this. This effector activity bioassay was a very useful tool for sensitive rapid sample analysis, indicating the effector function quite accurately. And even low levels of binding activity that we saw in the biocore can result in substantial levels of cell-based activity, but we've identified, we think, for the first time, a variant which is completely devoid of FC effector activity. And of course, that could be very useful for all sorts of different clinical applications for treatment of autoimmune diseases and other situations. And we've done a number of other experiments that I haven't shown here to show that the manufacturability and the stability, the glycosylation, the immunogenicity are really unaffected by these mutations. And I've shown you here that the binding to the FcRn and largely the pharmacokinetics in vivo are also unaffected, and these variants are very similar to wild type. So I think that's wraps it up. And I just want to acknowledge my colleague Ian Wilkinson, who came up with the idea for this in the first place, and also colleagues at Absolute Antibody who've manufactured these hundreds of variants for us, as well as at Antibody Analytics, who did some of the biocore analysis, Celintex, who did the cytokine release, and of course, friends at the JAX laboratory. Without them and without these unique mouse models, we would not have been able to do the pharmacokinetic studies that are so important. So thank you very much.